Welcome to today's webinar, the MMRF CoMMpass Study, Insights brought to you by the Multiple Myeloma Research Foundation and sponsored by AbbVie, Amgen, Bristol-Myers Squibb, Celgene, Genentech, Janssen, Karyopharm, Novartis, Takeda Oncology. It's my pleasure at this time to turn it over to Mary Jerome at the Multiple Myeloma Research Foundation, who will begin the webinar.
Hello, everyone. I'm Mary Jerome, Director of Medical Communications and Education at the Multiple Myeloma Research Foundation. Thank you all for joining us today for this webinar. I would also like to thank AbbVie, Amgen, Bristol-Myers Squibb, Celgene, Genentech, Janssen, Karyopharm, Novartis, and Takeda Oncology for providing educational grants to support this webinar. First, a little bit on the MMRF. The MMRF is a patient-founded organization started in 1998 by Kathy Giusti soon after she was diagnosed with multiple myeloma. As a patient-founded organization, we have a keen sense of urgency and a laser focus on extending patient lives. As many of you know, myeloma is a highly heterogeneous disease, which means every patient's disease is different. So, each patient should receive treatment specific to his or her type of myeloma. That is the promise of precision medicine. With such a high degree of disease diversity, the MMRF is focused on generating and aggregating as much data as possible to capture the full picture of myeloma. The cornerstone of this effort is our CoMMpass study, which now has the largest genomics data set of any cancer. We make sure that data is available to researchers worldwide to develop hypotheses for new, more precise treatments, and then we push those hypotheses into clinical trials. This is our precision medicine model. We are already expanding our genomic data collection efforts beyond CoMMpass into relapsed and refractory patients. And recently, we announced several major multi-million-dollar investments that leverage this model. We have investments in initiatives in immune therapy, in myeloma prevention, and also in high-risk myeloma. You can read more about our model and our initiatives on our website. To follow all the latest research and educational updates, I strongly encourage you not only to visit our website, but also to follow us on Facebook and Twitter if you are active on these channels. For today's webinar, I'd like to introduce our two speakers. Dr. Jonathan Keats is the Director of Bioinformatics and an Assistant Professor in the Integrated Cancer Genomics Division at the Translational Genomics Research Institute in Phoenix, Arizona. He's also the Scientific Director of the Judy and Bernard Briskin Center for Multiple Myeloma Research at the City of Hope. His main research focus is on the genetics of multiple myeloma and other hematological diseases, and he leads a lab that concentrates on developing a better understanding of these diseases. Also joining us today is Dr. Sagar Lonial, who currently serves as Professor and Chair in the Department of Hematology and Medical Oncology at Emory University School of Medicine and Chief Medical Officer at the Winship Cancer Institute of Emory University. He is an internationally recognized authority in multiple myeloma treatment and research, and Sagar also serves as the principal investigator of the CoMMpass study. At the Winship Cancer Institute, Dr. Lonial treats patients with multiple myeloma and is a lead member of the bone marrow transplantation team and clinical trials team. He's board certified in hematology and medical oncology and an active member of several professional organizations. Today, Dr. Keats and Dr. Lonial will provide insights into the CoMMpass study and present some of its findings. The topics for today's discussion on the CoMMpass study include an overview of the MMRF CoMMpass study with general research findings, including genomic profiles, subtypes identified and what they mean, genetic mutation analysis and high-risk disease, and the clinical implications of the CoMMpass study. Before I introduce our first speaker, let me provide you with a brief background on the CoMMpass study. The CoMMpass study was initiated in 2011 and is a landmark study focusing on the genomics of myeloma. The goal of the study is to learn which patients respond to myeloma therapies and to achieve better treatments strictly targeted to each patient's biological makeup. In the study, newly diagnosed patients will be followed for at least 8 years. So you can see that we started enrolling patients in 2011, and we currently have enrolled 1,150 patients from 90 countries in the CoMMpass study. Enrollment of this trial was closed in July of 2015. And you can see there on the slide the breakdown of the countries where the study subjects are located, mostly in the U.S. We have a contingent also in Canada, some in Italy, and some in Spain. So to enroll in the study, patients were newly diagnosed, and their bone marrow samples were taken at the start of the study when they were newly diagnosed, and also when the patients responded to treatment, and also at relapse. And all of these samples were collected and analyzed. Throughout the course of this study, each patient is seen by their doctor at least every 6 months for the entire 8 years of the study. So you can see that we are collecting a vast amount of data. On this slide, you can see sort of a picture of the makeup of the subjects in the CoMMpass study. So you can see that the average age of the CoMMpass study patients is 64, but the youngest was 27, and the oldest is 93. So there's quite a range of ages. As far as the genders of the study subjects, 2/3 of the subjects are male and 1/3 are female, which really reflects the incidence of myeloma in the general population. And you can also see there the race and ethnicity of the CoMMpass study participants. 77% Caucasian, 2% Asian, and notably 16% African American participants, which is actually a very high percentage for a clinical trial. And here on this slide, you can see the type of data that we're collecting, along with all of the genomic and clinical information from patients. We're also collecting a great deal of data on the types of therapies that patients are receiving, and that will be paired with the outcomes that patients achieve while they're on these therapies. And we're looking at this information as first-line regimens, second-line regimens, third-line regimens for all patients. So all of this data collected clinically, along with the genomic information, will be utilized as we move toward more effective therapies for myeloma. So now I'm going to introduce Dr. Jonathan Keats, who's going to tell us more about the genomic component of the CoMMpass trial. Jonathan?
Thank you, Mary, and welcome to all the several hundred listeners out there. Appreciate you giving us an opportunity to give you insights into what we're doing in this really landmark study for patients with multiple myeloma. So I'll walk you through briefly what my team here at TGen is doing and try and show you at a simplistic or early level what we're really doing and how we leverage the data on a daily basis. So the first thing to understand, and Mary mentioned we're sequencing patients, is that when a patient who is participating in the CoMMpass study decides to participate, they provided a peripheral blood sample, which we use as their normal DNA, and they also provided a tumor sample through a bone marrow aspirate. And from that bone marrow aspirate, we actually have a procedure to pull out just the tumor cells out of that sample so that we can take those tumor samples and compare them to the patient's normal that we got from their blood. What we do with both of those two samples is we isolate DNA and RNA, shown on this slide. So DNA being a very big, long molecule, creating the chromosomes that really define what's different between each one of us. And then also the RNA, which is encoded within the DNA as what produces genes or the working elements of our cells, so the proteins that are produced. What we do on this side is from those isolated DNA and RNA, we fragment them up, so we take it and we break it up into little pieces. And those little pieces we add what we call adapters, which is pieces of DNA get added to the end, those red and green portions, which allows us to put those DNA molecules or little pieces onto our sequencing instruments seen there. And with these instruments, we basically slowly add, actually not very slowly now, it takes about 24 hours to sequence a genome on one of these instruments. We add a color that lights up each one of those dots, which is a molecule or one of those individual molecules, and there's millions or billions on the individual sequencers. And we can, with cycle after cycle, basically read the sequence of the DNA. So our DNA is made up of A, G, C's, and T's, and we can basically read each one of those molecules. After that's finished, it goes on to our high-performance computing infrastructure here at TGen, where we put all that information back together. So we broke it into little pieces. Now we stitch it all back together to really understand what's different between the normal cells and the tumor cells in each individual patient. So to give you an idea what that looks like, this is actually some of the true raw data that people on my team look at on a daily basis. So at the very top is what we would say is the control DNA from the peripheral blood, or the normal DNA of this individual. And what you'll notice there is it's all gray, which means it's boring and normal, and it matches the reference human genome. In this case, this tumor DNA, you see there's a bunch of T's where otherwise there really should be an A down you see at the very bottom. And that T actually not only shows up in the tumor DNA, it shows up in the tumor RNA. And what's really important there, and what is one of the unique features of CoMMpass, is that by sequencing the DNA and the RNA, we can separate mutations that lead to mutant proteins in tumor cells versus things that exist in the patient's DNA but do not really produce a protein that has a functional consequence within the tumor. It lets us to separate the signal from the noise. So in this case, this is actually a mutation that even has a therapeutic recommendation. So patients who have this T in this position actually have a mutation that's common in melanoma, and there's a drug specifically for that mutation that would allow us to do what we would call precision medicine on those patients and give them a specific therapy related to an abnormality that's in their tumor. So as Mary mentioned, this is one of the largest cancer genomic studies, and still, to our knowledge, is the largest in regards to total effort that's gone on in a single tumor. And if you compare CoMMpass, and these little acronyms at the bottom reflect several large studies that have been run by the National Institutes of Health here in the United States and through international consortiums around the world that have studied individual cancers with a similar type of technology. And what you quickly see is outside of the one breast cancer study run by the US government, CoMMpass far surpasses all of those studies in total amount of data generated. When we look at total patients studied, only that one breast cancer study has about 100 more patients studied than we have on CoMMpass total. So this is a monstrosity of a study compared to a lot of studies out there, and it really highlights the value of a patient-driven organization trying to say that regardless of how common this disease is, we're going to make sure the knowledge base of it is the best in class around the world. So what have we learned from doing all this? We've come across a lot of things, and there's been several hundred abstracts and numerous papers now already published on the results from this data set. We have confirmed and expanded a large list of the genes that are altered and play a role in myeloma, and we'll show you, comparing two previous MMRF-funded studies, how this study has taken us one step further. We're starting to really understand how the disease evolves and adapts to treatment, and we'll show you an example of one patient and how they change with time. And we are also learning a lot more about high risk, and particularly how physicians are communicating high risk today, and maybe some of the things that we would like this study to really change those communications in the future, and I'll walk you through each one of those. So I mentioned this thing called precision medicine, and what is precision medicine? And I can tell you today that Dr. Loniel at Emory does practice precision medicine because he knows characteristics of his patients today. He can see that they maybe could get a transplant or maybe couldn't get a transplant, or maybe have other things that define which treatments he would use. But in average today, if we consider what's typically presented today as evidence-based medicine, where we say for the majority of myeloma patients with this certain phenotype, we would give you treatment 1 because we expect that to work the best for that group of patients. But obviously that's not going to work for all those patients. It works for a large percentage, the biggest percentage of patients. Should a patient not perform well with that therapy, then we would go to treatment 2. And then if not working on that one, we would go to treatment 3. And an example here of things that we're really trying to push with CoMMpass is these two patients here, one in the black dot, one in the red dot. And if we move over to a precision medicine model and we say, well, if we know the patient who is that black dot will ultimately only respond to treatment 3, why take them through the effort of testing treatment 1 and then testing treatment 2 when we might know that they would respond best to treatment 3 to start with? And those are probably some of the earliest things that are going to really come out of CoMMpass, is identifying patients where we can give them specific groups of myeloma therapies that exist today, but they might be optimally targeted to that patient to get the best response, but with the least side effects for the patient. And then there's the patient who is in red, where today we would say they're probably not going to respond to any of those core types of treatments. But maybe as an example we had before, there's something unique to their tumor where we could give them a drug specific to their tumor to try and give them something that wouldn't really be given to the average patient, but will be very beneficial to them, we would hope. So that gives you an idea of the precision medicine model that we're pushing. Why is that so important? And this is an example of, we take all the RNA data that we have for the over 1,100 patients today on CoMMpass and just ask, is there different patterns within these patients? And we've known about this in the disease for a while, but I think what CoMMpass has really done is really cemented the number of patterns or types of myeloma that are out there. And in this case, with the largest data set out there on myeloma, we would really say that there are 12 really distinct types of myeloma phenotypes. So when a pathologist looks at a cell, the tumors under a microscope, and diagnoses multiple myeloma, all these patients are told they have multiple myeloma, but there are very distinct types of myeloma today that we know of, but generally we treat all of them the same. And obviously what we're hoping to find with CoMMpass is that there are certain groups of these patients who respond to certain treatments better, and that way we go back into that more precision medicine model and move certain therapies up in the order for specific groups of patients. I did mention we're starting to really understand the genes that are important in myeloma, regardless of what subtype or type of myeloma you have. A lot of these mutations exist in all those different groups. And what we've seen in the past is, we did a study about 5 years ago that, or more actually closer to 7 years ago now, what we called the Multiple Myeloma Genome Initiative. And in that we sequenced 200 patients, not the 1,000 plus we have today. And we identified all these genes that are circled in red as being important in myeloma. What we've now added is, with the CoMMpass cohort, is all these additional genes that are not circled in red, we've identified as being important in the disease through this additional data set, which includes a much more comprehensive view of the genome than we had before, but also larger patient populations. So things that are rare are now being identified in this population. And one of the things that I'd like to take you through that we've really championed in this disease today, and regarding the complexity of the data we've collected in CoMMpass, we've been able to separate out something that's really important to how we communicate to patients. And the simplest explanation to talk to this, and this is going to be related to a lot of patients out there will have testing for deletions of 17p, or sometimes they've heard of it as the p53 gene, if it's deleted or not. And what we've seen looking at CoMMpass is it's really important to realize that every one of us, when we're born, we have 2 copies of this gene, p53, which when it's working, we can equivalent it to the brakes on your car. It's considered the guardian of the genome and the gene that prevents cancer. So in some patients, we know they have a deletion of one of these arms, and they would basically show as if the front brakes were working, but the back brakes were not working. And what we've been able to do with CoMMpass is not just do that really simplistic measurement, but actually get into, say, there are 2 copies of the gene. Can we actually separate out if a patient has the front brakes working, but not the back brakes, front and back brakes, or if they have no brakes at all, the back brakes and the front brakes are gone? And this is really important in the change in the results, and also how we, in the future, we can talk to patients. So today we just, in the clinic, test to see if the gene is deleted. And if a patient has this gene deleted, they're told that they have high-risk disease. What we could separate with CoMMpass is to actually say, since patients have 2 copies, it's really important to know if they still have 1 copy that's working, or if both copies are completely gone. And that dramatically changes the outcome of patients. So the patients who have both copies lost are doing poorly compared to all other patients, while the patients who are listed as having a deletion or a single mutation are doing just as well as those patients who have both copies present. Why this is important is because obviously today, all the patients that have one of those events are being told they're high risk, and we're telling about 10% of patients they're high risk, when really it's only about 3% of those patients are truly high risk, and the other 7% are getting a bit of misinformation. And obviously this is kind of the hope that with CoMMpass we can identify these differences. We can encourage hospitals to do this more complex type of testing, which is available at many medical centers today, and then to join the data types together to make this kind of interpretation so that they get the right information to the right patients correctly. And why does that become important? And this is just an example, again, from CoMMpass, taking two of our patients who otherwise would seem like they're very similar. They're both females. They're both about early 70s. They're both the same ethnicity. They have the same type of stage of myeloma. They got the same treatment. By any conventional testing that would be done in the average hospital in the United States, they looked very similar. But one progressed in just over a month, and the other one is well over 18 months without having progressed. And actually, that patient is now over 28 months without having progressed. If we look at that and we just add that extra status of is the gene also mutated, so what we see is the first patient who progressed very quickly had a deletion of 17p, which was picked up in the hospital, but also had a mutation that was not picked up. And having both copies or both breaks cut, this patient progressed very quickly. The other patient was told they were high risk, but actually has had a very good outcome on a fairly standard treatment regimen that's out there. Now, the next thing that totally differentiates the CoMMpass study from all those other studies that I mentioned at the onset that have been done in other cancers is, as Mary mentioned, over the 8 years of observation, when a patient progresses, we get another sample from their tumor, and it allows us to start asking questions about how the tumor has changed with time. Do certain therapies change the tumor with time? And how those things interrelate so we can inform clinical decision-making better. And what's really important in this slide is just you'll quickly see that a lot of these patients are put into specific groups at diagnosis or baseline. But when they progress, they either stay generally on the same group with time, or a lot of them are collapsing into the group, the second from the bottom, that we call the PR, proliferation group. And this is a group of patients who have very poor outcome when they're diagnosed and they have that phenotype. But also if you progress into that phenotype, again, these patients tend to have very poor outcome. And what we've been able to do with CoMMpass is start to identify the things that have changed with time to allow causing the patient to change groups, which also gives us the opportunity to start monitoring in advance for these kind of events to occur so that we could predict which patients are going to have an aggressive relapse in the future. That will, again, allow physicians to potentially propose new clinical trials and increase monitoring efforts. And just as an example of that, if we go to the next slide, is just taking one of these patients, and you can see a little bit of the really detailed data that we generate on each one of these patients. So starting on the right-hand side is looking at the copy number of the patient. So the center red line is what's normal, and whenever the data is above the red line, it means we have extra copies of that DNA region, and whenever it's below the red line, means we've lost copies of that DNA region. And if you see the arrow that's pointing there on the bottom progression sample, there's a little tiny red dot, if you've got eyes better than mine because I can barely see it. There's a little red dot that is the event that we think is causing this patient to change from that group, which we call PRL3, into the progression group at progression. And if you look on the bottom left-hand side is actually some of the raw data from the whole genome sequencing data that we do on each and every one of these patients. And what you'll notice at baseline is there's just a lot of gray bars and little color dots that you can see there. But on the progression side, you see these big red bars. Those are deletions that did not exist or were not detectable at baseline that are present at progression that have knocked out this gene called CDKN2C, which is basically the brakes on cell division. When it's absent, the cell just keeps dividing. It doesn't have brakes, so it keeps dividing, going and going and going. So this is another one of those examples where losing 2 copies of a gene that causes brakes on cancer creates a very specific phenotype that we can now track with CoMMpass, and now we can develop assays to monitor for patients who have these events. So just to give you a summary of a lot of that kind of detailed information, this is one of the largest studies out there on any kind of cancer, and is by far one of the most systematic studies, having clinical data and longitudinal data on these patients. It's a huge wealth of data for the field that everyone greatly appreciates. It allows us to identify the genetic mechanisms that drive the cell growth and survival. It allows us to identify things that contribute to drug resistance, and it's important to note that there are a lot of new drugs that are highly effective in myeloma that we're already starting to have patients seeing those new drugs within the cohort. It may also allow us to predict which patients will respond to which therapies, and ultimately, again, with the goal of developing a more personalized approach to myeloma therapy. So with that, I'll pass it on to my colleague, Dr. Lonial, to give you an overview of the clinical data within the cohort.
Okay, this is Mary. I just wanted to add a couple of comments to the end of your talk, Jonathan. And I think that it's just amazing what you pointed out, that via the generosity of the patients who have consented to participate in the CoMMpass trial, we now, in myeloma, have the largest genomic data set in all cancer. And I think that's amazing. And we thank our patients for enrolling in this trial. It's so fascinating that we've been able to, at this point so far, determine that there's at least 12 different types of myeloma, and I'm sure that we'll find more of those in the future. We're beginning to help define who exactly is a high-risk patient in myeloma, which is so important to be able to identify those patients up front so that they can be treated appropriately for the level of risk of their disease. And finally, through this data, we're beginning to identify genes that are commonly mutated in myeloma throughout all different patients. So this is really going to help us in the development of precision medicine approaches in myeloma, which will benefit patients since they'll be able to get treatments that are specifically tailored to their disease. So now I'm going to introduce Dr. Lonial, who's going to talk to us more about the clinical implications of the CoMMpass study data set. Sagar?
Thanks very much, and I appreciate everybody's attention, and I think Dr. Keats did a great job setting up a lot of the really important and baseline work that we've done in CoMMpass that allows us to begin to ask many of the questions that we think are really critically important to helping to really make a difference in this disease over time. So I think there are a number of different questions that we've already begun to ask in the context of available data. And I think that there are a couple of things to remember about CoMMpass. First is that the follow-up is actually relatively short. So many of the real long-term benefits we're going to get are going to take a little bit more time for us to see how patients evolve in terms of their treatment over the next few years. But already with some early follow-up data, we've seen differences between certain types of patients based on their mutation status. Second questions are going to be, how do we define high risk, as Dr. Keats already addressed, the role of transplant, and questions that many of us now, I think, are beginning to accept as relatively standard that have not necessarily been wholly adopted in the community, and that is the role of triplets versus doublets for newly diagnosed myeloma patients. So let's begin with a couple of these, because there is some data that I think we can pretty clearly say from CoMMpass that has now since been validated in phase 3 trials, and that is 3 drugs are better than 2 at the time of initial diagnosis. And we initially had shown in CoMMpass almost 2 years ago that patients who took 3 drugs at the time of initial diagnosis had a better long-term progression-free and even overall survival than patients who took doublet-based therapy or 2 drugs at a time at the time of initial presentation. This data was then validated subsequently by a couple of large randomized phase 3 trials that also demonstrates improvements in PFS and overall survival. And so I would argue many of the insights we gained from the way that patients were being treated in the CoMMpass trial actually yielded some of the evidence we use to make that practice change over time. The second one is an endpoint that I think is really important. And one of the big buzzwords that occurs right now is, can we do this instead of transplant? Can we replace the role of high-dose therapy in autologous transplantation? And I think that there's a lot of confusion, particularly among docs that are not at big myeloma centers, as well as potentially among patients, about the potential role of high-dose therapy and transplant in an era where we're using new drugs and modern drugs. And what I think CoMMpass really helped us to identify was that very clearly there continues to be a role and a benefit for transplant across the board, and that patients who have a transplant stay in remission longer, and at least with short follow-up, it's suggestive that their overall survival is better as well. So while there may be a lot of discussion about the timing of transplant, transplant clearly does offer continued benefit for patients. Now, what about some additional analyses that are currently ongoing in the context of this data set? Well, one that I think is really important to look at is data on differences in racial disparity and myeloma deaths. And the reason that this is so critically important is we know the frequency of myeloma is much higher in an African American population. It's almost double compared to a non-African American population. The age with which myeloma presents in an African American population is about 5 to 10 years younger than it is in aggregate for patients across the board. And as Mary pointed out early on, CoMMpass has about 16% of patients that are African American. So it does give us the opportunity to really look at a relatively large data set at not just outcomes, but differences in genetic outcomes over time as well. We also know that there may be some barriers of care in terms of the ability to get care delivered, and so it's not clear whether those things play into different outcomes for patients who are African American versus non-African American myeloma patients. Now, one of the things that we did early on, and a couple of other groups has done as well, is begin to look at the difference in genetics and genomics between European Americans and African Americans. And one of the things that really jumps out to you is that many high-risk features are seen in much lower frequency in African Americans than non-African Americans. And that actually suggests that there may be differences in how patients get to the diagnosis of myeloma. So, for instance, the incidence of P53 mutations is significantly lower in the African American population compared to others. And there were other differences that suggest a higher frequency of what's called hyperdiploidy. Hyperdiploidy is extra copies of odd-numbered chromosomes, and we know that in myeloma, isolated hyperdiploidy actually has a better long-term outcome. And in fact, hyperdiploidy appears to be represented in a higher frequency among African Americans and non-African Americans, suggesting that if you're an African American, perhaps your genetic risk in the context of myeloma may actually be better than it is for non-African American patients. And when we went back and looked at CoMMpass, it looked like treatments were actually relatively similar between the African American and the non-African American patient populations. And I think that's really important because it takes out the access to care or ability to receive treatments as a variable in terms of outcomes. And if you remove that, what you can see is no significant difference in progression-free survival and overall survival. And what that suggests is actually somewhat different from larger population-based studies that don't take necessarily into account access. In CoMMpass, we can say everybody had good access, and in fact, the outcomes were quite good, suggesting that if you can make access equal across the board, you may be able to raise the level of outcomes for patients who are an African American in descent. And so I think that's a really important observation. It does certainly, it's a new data set that we didn't have access to until we did CoMMpass, and I think partnering it with the genomic and genetic data really allows us to make some very powerful and important conclusions over time. Now, what about other analyses that we've done? There was an analysis done looking at the impact of mutation load in terms of outcomes, and we know for other diseases, the more mutations you have, the shorter the progression-free and suggesting the shorter the overall survival may be. And this was actually proven in CoMMpass as well. In red, you can see patients with lower mutation burden. In blue, patients with higher mutation burden, suggesting that more mutations is associated with higher risk disease and maybe a poor risk feature as well. Now, what are some of the ongoing trials and future directions that we're doing within the MMRF and the MMRC to really try and address additional follow-up on some of the findings from CoMMpass? Well, the first is a molecular profiling initiative. And this is a really exciting opportunity for patients who were seen at MMRC sites to have free genomic profiling performed as part of routine care. And what happens in that situation is that patients provide a sample of their bone marrow, which is being done for another reason perhaps. It gets sent up to the University of Michigan, where they do a CLIA-certified sequencing on them, and then it potentially offers them the ability to go onto a trial that we're really excited about, and that is the MyDrug protocol. And so I'm going to show you the MyDrug protocol on the next slide. But as you can see here, the molecular profiling protocol is basically across all the MMRC sites. The goal is to enroll 500 patients with relapsed and refractory myeloma to further catalog the pattern and types of genetic mutations that we see. And again, in some patients, we've identified potentially targetable mutations. Now, this is really important and I think leads to the importance of the MyDrug protocol. And the MyDrug protocol, as you can see, is an umbrella protocol being run within the MMRC, or the Multiple Myeloma Research Consortium. And the reason it's such a critical protocol is that we know that there are certain mutations, like, say, RAS, or IDH, or CCND1, or PI3K, AKT mutations, but we don't know whether targeting that specific mutation actually makes a difference in myeloma. In other cancers, there's some data to suggest if you've got a specific mutation that taking a drug that targets that specific mutation may have a role in temporarily controlling that disease. In myeloma, it's not as clear because, as you heard from Dr. Keats earlier, there are different clones of myeloma within the same individual patient. And just because you see a mutation in the RAS or RAF kinase pathway, that doesn't mean that using a drug to target it will have an impact on the disease. And so while the identification of mutations is clearly important to helping us get to a precision medicine field, we need to test that hypothesis to prove that it really makes a difference. And that's what MyDrug is all about. MyDrug is about testing those individual mutations with specific drugs targeting it and seeing whether in a larger patient population that drug mutation pair actually makes a difference. And the reason I say that is right now we cannot say that with any level of confidence. There are anecdotal reports that targeting common mutations such as BRAF may have an impact on patients with myeloma, but the data in aggregate does not show a significantly high overall response rate or long duration of response for those patients. So we need to understand who's best suited for that targeted approach, and if they're not best suited, why, and what can we do to enhance the response rate of those patients over time. And to get to this, the MMRF has now created the Answer Fund. And the Answer Fund is as you can see, a 3-year, $10 million crowdsourcing project where projects are elicited to use data within CoMMpass to answer questions that are relevant and important to patients. And there were 2 Answer Fund grants that were given out in the last 2 months. One was given to Dr. Boyes from our center here at Emory, and the other to Dr. Mitsiadis at Dana-Farber. And these are the first of what we hope will be many Answer Fund grants that are required to use CoMMpass data as part of their strategy or hypothesis for why this grant proposal can really make an impact on patients. And I think that, to me, is really the heart of the question. We have data from genetics and genomics on 1,000 patients. How do we use that data not just to answer some additional scientific questions, but that those scientific questions translate into changes in patient care? And I'll give you an example of some data that Dr. Boyes put together as part of his Answer Fund project. This is uncovering a new way to define high-risk myeloma. And in this analysis, he and Dr. Barwick, who's also at our center, looked at a specific translocation in patients with myeloma. And we know that translocations occur quite frequently. About 2/3 of myeloma patients have some sort of a recurrent translocation. But what was identified was a translocation into the IGL region. And the IGL region is the lambda light chain region of the immunoglobulin gene. And the reason this was important is that IGL translocations tend to occur in the hyperdiploid group of patients. And so a group of patients that we may previously identify as a good risk subset may actually have very poor risk disease. And we wouldn't have known that without CoMMpass, because no other sequencing protocol that's being performed in a large number of patients would have had the ability to identify this IGL translocation the way that CoMMpass was able to do. So basically, this observation would not have occurred were it not for the comprehensive way that Dr. Keats described we were doing sequencing in the context of CoMMpass. Really powerful data that would not have existed in the absence of this trial. So how can patients help tailor their therapy? Well, again, I think it's important to understand what are the potential options out there. The role of true precision medicine in myeloma is one in evolution. Just because you have a mutation and we have a drug doesn't necessarily mean that that drug will help that patient with that specific mutation. But it may allow you to participate in a clinical trial where we can really begin to answer that question, and trials like the MyDrug trial will really be critical to helping us to answer that question. Additionally, getting access to more patients with relapsed and refractory myeloma in terms of the relapse sequencing initiative that I described that's being run through Michigan is really critical to long-term success of our data collection piece that will ultimately continue to have impact on patient care. And again, participation in the MMRF Cure Cloud allows us to track those outcomes over time in patients with known mutation panels. So, I think in terms of some quick final thoughts, and then I'll turn it back over to Mary, it is clear that a one-size-fits-all approach may be inappropriate for certain types of myeloma. We know, for instance, that high-risk myeloma should be treated differently than standard risk, particularly in the context of maintenance therapy, and understanding how mutations can provide additional information for this discrimination is really key. We know that patients are different in terms of how they get to myeloma. African Americans may be different in how they get to myeloma than non-African Americans, and using that data to understand disease pathology is really important, but it may actually have a role in prognosis as well. And really the goal is participation not just in clinical trials that have an impact by using a treatment, but also clinical trials that collect genomic data before treatment and after treatment to allow us to understand the impact the specific treatment has on the types of mutations that given patients have during the treatment process itself. And with that, I think I'll turn it back over to Mary and let her do some of the Q&A.
Okay, great. Thank you, Sagar. That was so a great summary of what's happening with the CoMMpass clinical data. As we talked about in the beginning, this study has been ongoing since 2011, and it does take quite some time to collect enough data to be able to really say with certainty what's happening in patients as we analyze it. But it's great that you pointed out that now we can say with some certainty, thanks to CoMMpass data, that we know that a triplet regimen provides better outcomes to patients than a doublet regimen, and also that transplant is still advised to patients versus not having a transplant based on their outcomes that we see in CoMMpass. Also really interesting, the differences that we're seeing in patients genomically and clinically due to their ethnicity, and how their access to treatment may have impacts on their outcomes. So we'll definitely be looking to see more information on those studies. And I think it's super important. You pointed out that it's really vital that we utilize the CoMMpass data to make an impact on patient care. And that is really what we're working toward here at the MMRF and through all of our MMRC sites and our CoMMpass sites, is to really gather this data so that we can make a positive impact on patient care. So now we're going to switch over to the question and answer session. As a reminder to the audience, If you'd like to ask a question, type it into the Q&A box and click Enter. We're going to be answering questions in the order in which they were received, but of course, there's a lot of questions and not all that much time, so we may not be able to get to answer every question that's come in so far. One of the questions that somebody asked I thought was a really very insightful question, Jonathan. This one goes to you. They asked, how does, when you take someone's bone marrow and do sequencing, how do you determine the clonal heterogeneity of the clones at the time of sequencing, and how might that affect the results that you get from the sequencing?
Yeah. So before sequencing the patient, we have little information on the heterogeneity. Now, one thing, and it relates to one of the other questions, I think is one of the most beautiful things about working on multiple myeloma as a researcher is the fact that plasma cells, which are the tumor cells, are rare. So when we sequence a bone marrow sample, we enrich the plasma cells out. So almost all the all the cells that we're sequencing are almost 100% tumor. Now, what we know is there is heterogeneity within that mixture and the types of events that may be present in 10% of the tumor cells or 50% of the tumor cells. The way we've addressed that from the CoMMpass side is CoMMpass, and again, thanks to the really generous funding from the Multiple Myeloma Research Foundation, we've taken the depth of sequencing about twofold higher than most of the typical studies that have been done in the field, which gives us the sensitivity to pick up events that are in about 10% of cells with almost 100% specificity. If an event was lower than that in frequency from a mutation, we may not see it, so it'll be hit or miss. But obviously, if we get the same tumor from that patient later at a relapse and that event has actually provided a competitive advantage to the group of cells that had it, we would expect to pick that up at a later point. And that's kind of how we deal with it from a CoMMpass side, was twofold. One, sequencing is very deep to have sensitivity, but then also by picking up the samples with time so we can detect those kind of changes if they really do have competitive advantages.
Okay, thank you. I'm going to ask Sagar this question, and this actually came in from a number of folks who are listening into our webinar here. And I know that there is some controversy surrounding this in the field, but several people wanted to ask what actually defines a patient as being high risk. And I know there's a number of factors there, but could you go into that a little bit, Sagar?
Yeah, and I would tell you to stay tuned to a paper coming from the International Myeloma workshop that I'm writing that is actually describing the way the small panel of people at the India workshop define this. But there are lots of different ways you can use to define it. So the use of FISH testing, for instance, that is looking for things like 17p deletion, 414 translocation, 1416 translocation. There's some data sets that say that extra copies of chromosome 1q is associated with poor risk. Those are the simple, easy ones that are done, I think, on most FISH testing panels. Other criteria that can be used are things like gene expression profiling, where there is high risk and standard risk that's defined by the expression of a number of genes within that GEP definition. There's also things along the lines of mutation sequencing that were published by the group from the UK that may or may not have some utility, and right now are not quite ready for prime time, I don't think yet, until the CoMMpass data really gets a little bit more mature to help us confirm some of that data. There's also clinical criteria, things like circulating plasma cells. If you've got lots of plasma cells in the blood, that's not a good thing. And then the other piece is what we call extramedullary disease at the time of presentation. Patients that present with those lumps or bumps that are not necessarily in bone, but are in lymph nodes or other areas like that, that can be considered a poor risk feature. A high LDH, which is a blood test that we look for. Those are some clinical criteria that we use to define high risk as well.
Great, that's really, really laid that out really nicely, Sagar. Jonathan, I've got an interesting question for you, and this was sent in by one patient, but I'm sure that other people would be interested in this. So they're asking about commercial DNA sequencing services, such as 23andMe, and whether that type of service would provide any genomic information as far as myeloma is concerned. So can you sort of compare what that sequencing means versus the type of sequencing that's done in myeloma, the types of information that come out of those two different sequencing types, and then whether or not you recommend every patient should be genomically sequenced?
So in regards to some of the direct-to-consumer vendors like 23andMe and Ancestry.com, what they're providing are SNP chip analysis, so it's not true DNA sequencing, where they're looking at things that we know exist in the population, so they can genotype people at all those positions and know if you have an A or a G or a C or a T at that given position. That's almost exclusively done on normal cells, so it would be similar to our normal genome sequencing we're doing in CoMMpass, where you can do things like very exquisitely say the genetic ancestry of the individual, that this person originates from an Icelandic population, for example. Obviously, and I don't know the specific ones, but I would suspect that there are some of the GWAS or studies that we've done in myeloma. So these are genome-wide association studies where we identified that if you have an A or a T or a C at this specific point in the genome, your risk of getting myeloma is higher. A lot of those services will report on some of those events. I must admit, I don't know if those are being reported or not by 23andMe. I've never seen one of those for a myeloma patient with one of those events. But I think those would only at the very most give you something that would say you have a 1.1% more risk of having myeloma than another individual. It doesn't test any of the genetic things in the tumor cells that we would focus on as predicting your potential outcome as a myeloma patient. And then in regards to should everyone have their genome sequenced, I think the simple answer today is probably no, because it wouldn't dramatically change your treatment today. Most of the treating physicians wouldn't necessarily know how to leverage the information. If you're at a site, though, that is running a clinical trial that would do something like my drug, where we would start identifying treatment specific for patients and potentially altering your treatment, then I would definitely encourage patients that those types of treatment are asking to participate. You may not get onto one of those specific arms like a my drug because you don't have one of those events, but it creates an opportunity for you to have that kind of information. Obviously, more and more sites, the big academic centers in the country are doing tests of this nature locally to get a lot of the really critical data that's kind of pan-cancer is quite common, and many patients probably have that and may not even necessarily know.
Thank you. Sagar, we had someone who sent in a question regarding triplet treatment. In that triplet treatment is shown to have better outcomes. However, how do we address toxicity and the ability to sustain multiple drug regimes in the face of toxicity and side effects?
Yeah, I mean, that's always a balance. Every combination is not equally effective for every patient. I think the principle is that when you hit a disease with more than one drug that targets different mechanisms, you're less likely to have tumor escape and more likely to suppress the disease for a longer period of time. And that's the theoretical reason for 3 drugs versus 2, and it has borne out in clinical trials. The what to do with toxicity question is always one that is an important one to weigh, and that means that the dosing of the drugs may need to be adjusted in order to reduce the amount of toxicity that patients experience. Remember, for many of these trials, the goal is to give 3 drugs for 4 cycles of therapy, collect cells, and then move on to transplant. And so in that situation, the chance of long-term toxicity is lower than it is if you're going to give drugs for an extended period of time. which is what many who opt to delay or not do transplant at all do. And so figuring out how to balance the duration of therapy with dose adjustments and management of side effects in the short term really are the two things that people that see a lot of myeloma patients are able to do because we have so much experience with it.
So here's an important question that came in, Sagar. From a patient's perspective, does participation in the clinical trial risk the patient's overall survival period as opposed to those who follow a more standard treatment regimen?
You know, I mean, the reason it's a trial is that there's always a question that you're trying to answer. If it's a randomized trial where you're getting a new treatment versus a standard treatment, then I would say it doesn't necessarily risk the survival because you know, on the one hand, you're going to get what most people would get anyway, and on the other hand, there's a pretty good data set that speaks to the potential advantage of adding that other drug in. But the flip side of that is the data that we saw with the pembrolizumab trials where there were certainly early deaths that were seen in the experimental arm that may have been a fluke of that specific trial as opposed to something specific to the PD-1 or PD-L1 cascade. So anytime you're in a trial, nobody can guarantee your outcomes, but the goal of that trial is to try and advance the field, and trials are watched very closely from both the local level at the IRB, at the investigator, at the people that are enrolling and controlling the trial, as well as at a larger level at the FDA, as well as at the company, if it's a company-sponsored trial, to try and minimize that risk for patients over time.
Great. And since, Sagar, you have such a good overview of the entire myeloma field, one patient asked, how many myeloma trials are currently being conducted in the U.S. and worldwide? And how is the data from those trials shared, and is it shared?
That you can go to clinicaltrials.gov and put in the word myeloma and try and see how many trials are registered at the government right now. My guess is it's probably close to 1,000. I could be off by a factor there one way or another, but there are lots and lots of trials that are being done, particularly in the U.S. And the way that data is shared is twofold. The first is that at meetings like ASH or ASCO or the European Hematology Society or the International Myeloma Workshop, trial data is presented in abstract form to try and update all of our colleagues on what's out there and what we know about trials at a given point. The second is publications. When a trial has completed or done what it's supposed to do, there's then a manuscript that comes out from that trial that further informs people across who has access, anybody has access to those journals, to give you the results of that trial as well. So I think the ability for investigators to know what's going on is pretty good, and patients have access to much of that data as well.
Great. So I think we're just about at the end of our question and answer time, and I have one more question that I wanted to ask both of you, and this was posed in an interesting way by somebody who wrote in. And they said, can you provide an estimate of how far along you are on this quote-unquote trip toward finding personal therapies or precision medicine? So, for example, if you were traveling from New York City to LA, how far along are you on this trip in understanding the genes and what roles they play in myeloma and how a personalized treatment might work? Jonathan, you first.
You're going to be a United States Challenge geography guy. But we're not going to detour through Canada. My general thing is, obviously, I think the problem is if you're trying to say all myeloma patients or for specific genes, I think there are a couple of specific events. Maybe the closest one today that Dr. Loniel's team's really focused on has been those patients with the 11-14 translocation and potentially the use of venetoclax. I think that's something where we're way past St. Louis potentially, and I'd be interested to see what he would say for that group. And then obviously we have other examples, again, The one that the field has kind of worked on, but we haven't really as a field done great, is the 7% of patients with BRAF mutations. We might have got to Chicago, we might have got to Denver, but now we turned around and gone back to Chicago trying to find our way to LA. So I think for some of them, they're further along than we'd like. I don't think any of them have gotten out to us in Arizona yet, would be the unfortunate speak.
Great, thank you. And Sagar, last word. Sorry. Go ahead.
No, I think I would agree with Dr. Keith's assessment. I think with 1114, I think we're well beyond the Rockies, and we know that there's a certain subset of patients that clearly do well with venetoclax and can have very long and durable responses. But the question is, who else besides that subset may benefit from venetoclax? And there may be biologic reasons why that may be the case, and we're trying to understand that. With all of the other ones, including BRAF, I think it's going to require efforts like my drug to really get us closer to the West Coast. Because right now we have a lot of data, and we know the frequency with which these abnormalities occur in patients, but we don't know what they know in, say, lung cancer or breast cancer, that if you've got an EGFR mutation, your response rate with X drug is 50% or 30%, because we just haven't had an aggregate trial to really ask that question. And that's what my drug is all about. And so I think continued sequencing efforts as well as trials to test specific mutations with specific drugs are the only way we're going to put more miles on the car to get further along the road.
Great. Thank you so much. And that's all the time we have today for questions. On behalf of the MMRF, I'd like to thank Dr. Jonathan Keats and Dr. Sagar Lonial for their presentations today, excellent presentations describing the CoMMpass study. I'd also like to thank everyone here for joining us on the webinar. And finally, I'd like to thank AbbVie, Amgen, Bristol Myers Squibb, Celgene, Genentech, Janssen, Karyopharm, Novartis, and Takeda Oncology for sponsoring this webinar. So here you see that we have a couple of webinars upcoming. We're starting our always interesting immunotherapy webinar series soon. We have one coming up in June and one in July talking about antibody therapy and also CAR T therapy. So we encourage you to tune in for those. You can register for these on our website. We also have a number of upcoming live programs, multiple myeloma patient summits, which are educational programs. We have one scheduled for September in Nashville and one in October in Washington, D.C. Again, for more information on these programs or to register, please visit our website. And if you have additional questions, if you need some advice on your disease or any other resources that you are in need of regarding myeloma, please don't hesitate to call our MMRF Patient Support Center. You can speak directly to our MMRF Nurse Patient Navigators, and they can guide you to whatever resources and provide all the information that you need regarding our disease. You can call them at 866-603-6628. And again, we do have quite a lot of information on our website regarding myeloma and the initiatives that are ongoing at the MMRF and our clinical trials. So please stay connected to us via our website and also through Twitter and Facebook and other social media channels that we are on. For everyone who has participated in the webinar, we would like to let you know that you'll be able to view an archive of this presentation on our website when this is posted, and that'll be up within at least a week. And you will receive a link in an email to the archive program that you will receive. So, and we'd like to thank you once again for your attendance on this webinar, and we hope that you enjoyed it.